Materials and reagents used in Sds-Page are acrylamide solution and bisacrylamide for polyacrylamide gel, tris-hydrochloric buffer for stacking and resolving gel, SDS, sodium dodecyl sulfate, ammonium persulfate APS or TMAC to polymerize gel, 10-time running buffer or 1-time running buffer, Lam-Lee sample loading buffer containing SDS, glycerol and tracking dye, reducing agent like beta-mesactoethanol, protein sample, protein ladder or marker, gel stain like corrosive brilliant blue, D-stain solution, and bromophenol blue. Apparatus used in Sds-Page are vertical gel electrophoresis unit, gel tank or chamber, power supply with lid or clip, gel casting stand, glass plate and spacer for gel plate, combs to form well, electrophoresis buffer tank or chamber, pipette and pipette tip, microcentrifuge tube, gel staining tray, and timer.
Sds-Page, sodium dodecyl sulfate polyacrylamide gel electrophoresis is a widely used laboratory technique in biochemistry and molecular biology. It is used to separate and analyze protein based on their molecular weight. In this method, proteins are treated with SDS, which denatures the protein and gives them a uniform negative charge. The proteins are then loaded into a polyacrylamide gel and subjected to an electric field. As the protein moves through the gel, smaller proteins migrate faster and further toward the positive electrode than larger ones. Sds-Page is commonly used to estimate protein size, assess sample purity, and analyze protein composition.
First, sample preparation. A loading buffer containing SDS, beta-mesactoethanol, bromophenol blue, and glycerol is added into the protein sample. Protein sample may be biologically derived, for example, from prokaryotic or eukaryotic cell tissue, viruses, environmental sample, or purified protein. When the sample buffer has been added, the sample are then heated at 95 degrees for 5 minutes.
So here's an additional information about SDS. So actually, proteins are large biomolecule made up one or more long chain of amino acid residue, while amino acids are organic compounds that contain an amine group, carboxyl group, and a side chain. So proteins are formed when amino acids are linked by peptide bond and fold into one or more specific spatial conformation due to an interaction such as hydrogen bonds, hydrophilic interaction, disulfide bond, and ionic bonds. SDS denatures the native proteins by disrupting the hydrogen bond, hydrophobic interaction, and ionic interaction, while the reducing agent beta-mesactoethanol cleaves the disulfide bonds. When a protein mixture is heated, SDS binds uniformly off to the protein, making an intrinsic changes of the protein negligible compared to the negative charge contributed to the SDS. And finally, this treatment converts folded protein into a linear molecule with a net negative charge, allowing them to be separated into a polyacrylamide gel based on their molecular weight.
For the gel preparation, we use separating gel with pH 8.8 and a stacking gel pH 6.8, both containing acrylamide, bisacrylamide, ammonium persulfate, and TMAC. The gel is produced by polymerization between two glass plates anchored vertically in a cassette. The separating gel is poured first.
Sample application. First, load the sample that contain protein ladder in the well. Then, pivot the other sample in the well.
So the sample contains a bromophenol blue, which is a dye used to visualize the sample in the well. Glycerol increases the density of the sample and used to make the sample fall to the bottom of the sample well rather than just flowing out to the mix with the running buffer.
After sample has been loaded, close the lid. Electric field is applied across the gel, causing the negatively charged protein to migrate across the gel away from the negative electrode toward the positive electrode.
Next is the staining step. After electrophoresis, the proteins in the gel are invisible, so staining is essential. The gel is soaked in a protein binding dye, such as corrosive brilliant blue. The dye binds to the proteins trapped inside the gel, so protein bands appear as dark blue bands. Why is staining important? It allows us to see the separated proteins clearly. Without staining, dispersion result cannot be observed or interpreted. Next is the screening step. What happens in this step? The stain gel is compared with a protein ladder, which is molecular weight marker. The position of band is examined. Smaller proteins will move further down to the gel, while larger proteins will remain closer to the top. Why is screening important? It helps researchers to estimate the molecular weight of proteins, checking protein purity, comparing samples between lanes, and confirming whether the target protein is present.